All right. Well, welcome again, everybody. My name is Quinn Tran, the Sales and Marketing Manager here at DRVision. Thank you for coming to our webinar today, in which we'll be discussing with you our work on virtual reality for life science research. So before we start or restart, I'm going to go over the agenda. So very briefly. So we'll do a brief introduction today. We'll talk about image visualization limitations currently, what virtual reality offers, and what we at DRVision have created. We'll also go through a short demo with you and have time for questions and discussions at the end. So this is our team, and the members who are with me in the webinar today is myself, Bryce Graft, who is a software engineer here, who's the drive behind a lot of the things you'll see today. Huyen Lai, who is our product manager, and Luciano Lucas, who is the VP of Sales and Marketing. So from here, Luciano is actually going to take over and start this next part of the presentation.
One thing, if you want to ask any questions, you can ask during the actual demo or during the slides now. Huyen is tracking the chat, so you will be able to answer some of the questions, or if they're too complex to answer in a text, we will answer at the end, okay? So thanks so much for joining us. This is the first time we do a webinar, and so you've joined us on our opening session. It's really a pleasure to be here. We have some, we think, pretty cool things to share with you. But before we go there, I wanted to tell you a little bit about DRVision technologies, because I think it's relatively unknown, but there is a lot of good technology here, and I'd like to share with you a little bit about the history. So this company, or the people in this company, a lot of them actually have been together for a long time. They were part of a company called Neopath that created the first and still the only automated pap smear test device. It's still being used today in the hospitals. And once that company was acquired, the members formed a new company called SVision, which was focused on machine vision, specifically using a lot of pattern recognition and machine learning approaches, not just for life sciences, but also for other applications like metrology. And I'll show you that a little bit later. The company has been in the same, more or less, location here in Bellevue. This is on the west coast of the US, and we are around 22 people now. So it's a relatively small company, but a lot of output. So for biologists, which is, I think most of you, and including myself and Queen, apart from Bryce, I think is the only one that does not know biology, but everybody else, the only pictures that will be somehow recognizable are the ones perhaps on the bottom right. All the other ones are not really biology related. They go from 3D metrology to X-ray inspection of wafers and crack analysis and so on. All of these applications have been developed by DRVision and deployed in other people's products. And so very often you don't even hear about where the algorithms come from or where the software comes from. You just see, for example, Nikon branding something that we have created. But we do have two products. One is SVCell for 2D image analysis, and another one is CLQuant, which is something that Nikon sells. It's a similar version similar to SVCell, but with some customization. We are very active in terms of research. We have a few grants active at any one time, normally no more than 2. And right now we have 2, one from the Department of Defense and another one from NIH. And they almost always focus on life science applications. And here's an example of the ones that have been funded and are currently funded by NIH. And this kind of research and development then leads into the products that we can put in the market, like SVCell for 2D time-lapse image analysis. Along the way, the company has patented a lot of its technology, and the biggest group of patents we have is in machine learning. There's 11 of them, and that's becoming very popular again with deep learning and so on. So we are very well positioned there. We have a lot of technology that we plan to bring to the life science market in the coming months and years. And even in April, we will already bring the first tool for classifying cells using machine learning, which we won't talk about too much today, but at the launch date in April, it will be one of the major things we talk about. And you can see there's a few other areas that we've been active on the patenting aspect. We have a few collaborators, of course, to do the biology, to do the research. We need to have good collaborators at top universities, and here's some of them. We have a few more, and the list is growing in recent months, and we expect that it will continue to grow, and we will be even more active in the R&D, bringing the enabling algorithms that we've developed over the last 20 years and marrying those with interesting biological questions that top researchers have in order to advance science, essentially. So for me, virtual reality is really just a natural progression from what we have been used to seeing and doing. If you go back a few centuries, there was, of course, no technology like today, and even the microscopy was very rudimentary. So all the top researchers could do was observe directly with a microscope and draw. And amazing work was done. Both Prokinji and Cajal did amazing work, and there were other people at the time that were doing groundbreaking science purely using hand drawings, which is quite impressive. Today, almost no scientist can draw like these gentlemen did at the time. But things have progressed, and technology has developed, hardware has developed, and with the advent of good or high-resolution TEM, we could start to see some of the nanostructures, and that really opened up the interest for the detailed structure in neurons and how neurons connect to other neurons. Here is a neuromuscular junction that we see here with a lot of vesicles, and that provides clues. Just the visual inspection of a data set like this, and this is a single slice, already provides clues about the biology. And more recently, Jeff Liffman and his team have been doing amazing work. I've seen a couple of years back now an animation of a small area of the brain that has been fully mapped on a serial section EM, and then it explodes. Probably some of you have seen this video, and it's just mind-boggling how much information there is in such a small amount of brain. So that is now where we are. We have data sets that could be EM or could be light microscopy data sets, and they are very large physically, especially for the fluorescence light microscopy data sets. Physically, they're very large, and they portray very complex biology. And typically, we have also lots of cells and lots of cell types. So the tissue itself or the sample itself is very packed. It's actually really difficult to visualize the interactions on a flat screen or on a picture like we have here. So this is where rudimentary or the current methods of interacting with data sets which rely on a flat screen start to fall apart, because it's actually really hard to make measurements and observations on a screen that doesn't allow you to dive into it. And we do have, of course, software like what we will show you today, and there are others before, that can render in 3D and allow you some level of navigation in 3D and even making some animations. But it's still very limited. And that's what we and others are now trying to solve using virtual reality as a way forward. What we now can offer with the VR solution is a very natural way to interact with the data. You'll see me in a moment, or Hoyen if he wants to do it, walk around, walk in and out of a sample. We have three samples. It's an embryo and two neurons. one of them with lots of spines. And the level of interaction is really very, very engaging also. We've shown this now to a few people, and when people put the headset and start to interact with their data sets, it's also very pleasurable, actually. It's fun, but also very useful to ask meaningful questions and try to interrogate this, make those questions and try to test the hypothesis just looking at the data from different angles. We do support volume and surface rendering, and we allow for the interaction with results. So it's possible to grab different parts of the data and actually compare them side by side, even if they're far away from each other. So that's what we'll show in the demo in a moment. But I wanted to invite Bryce to give you a little bit more detail, technical detail on the headset, the handles, and VR in general.
Hi, I'm Bryce Graff. I'm a software engineer at DRVision, and we're just going to give a quick primer on virtual reality. Everybody has a different experience level. So virtual reality basics turns a room into a custom environment, and the idea is that the user can move and interact seamlessly. It involves, it kind of creates a really intuitive environment, and it's one of the most natural ways to interact with 3D data. And you're seeing it pop up everywhere, and it's for a good reason, because finally machines and graphics cards are basically to a point where it's usable and actually useful for real data. And soon enough, you'll see the headsets coming out with higher res and 4K, et cetera, for eyeball, but it's not quite there. I'll go over what we have now. So research, education, surgery simulation, if you're not using VR, you're kind of behind the times. Like it's here, it's now, it's good. And all of our team has been really hyped about it. So I'm just going to go on. So VR space, typically you can use everything. You can use everything sitting down, but typically you want to be able to stand up, interact with the data, and get more involved. So typical setup, you'll see our setup behind us. It's about 6 by 6, and you're able to move around in any direction, and it tracks exactly where your head's looking at. It's super intuitive. So the headset itself has to be really high frame rate because it's simulating exactly the position of where your head is looking. So the current frame rate is 90 hertz, and that creates a smooth enough experience. And the headsets typically use OLED, and that's the same as your LCD panel, except it doesn't have a backlight. And so the contrast levels are very high, so it makes for a really good experience. Each eyeball has 110-degree field of view, and what your computer is doing is rendering 2 separate scenes per eyeball, and each it simulates the parallax that you have, so it creates a seamless experience, and it renders 2 separate perspectives. So currently, you kind of have a trail behind you with a big wire. It's not, I don't know if you can see my mouse here. You can't actually get rid of the cord until they come out with a wireless headset. And that came out, but it sold out instantly. So the cable falls behind you, and most people have that. So the controllers themselves are motion tracked as well in the space, and they know the exact position. There's a touchpad on the front, and it tracks exactly where your thumb is, and so that's really useful for different interactions. There's a trigger on the back, so kind of like grabbing. This is just the Vive. The Oculus Rift simulates more of a hand, so it simulates several different fingers, and it's also very intuitive. It's kind of been up for debate which is better, but the Vive has had better motion tracking out of the box. So most people are using the Vive primarily. So you have your grip buttons, your D-pad, your trigger, and it tracks exactly where the controller is in the environment. And it's also rendered in the environment, and some applications will actually change the change the controller model to something else, which is also pretty neat. Okay, so for motion tracking, a lot of people see VR and they're wondering, how the heck does this work? So there's these things called lighthouses, and you put them at opposing side of the room. anyways, these lasers sweep across on your headset, and the Vive itself has, Vive itself, it kind of looks like a golf ball. And these lasers, okay, here we are. Each of these right here, these are photosensors, and they project infrared light and move across the room from both sides. And depending on the time when that infrared is sensed, it calculates the distance between these two, and from there it calculates the exact position of where the headset is. And it does the same for the controllers. You can see that the controllers will have a little loop on them and also have these infrared sensors. The video itself was just showing the sweeps that hit these different lasers. You can kind of see this red area right here that'll project across onto the headset. So, thank you. So it's incredibly accurate. It makes a really seamless experience, because if it wasn't seamless, it would make you sick. So it's sub-millimeter accurate, and what that allows is for really good and immersive experience. And we've seen people that I had no idea what their proficiency would be in VR, and they put that headset on, and it's kind of like magic. And as soon as you let them go and do their thing, they're moving objects around exactly where they want to put them. It's really intuitive, because it's exactly like what you'd expect walking around in real life and moving things around. So I'm just going to go over a quick detail of our different controls for our current beta. So the main control that you want to set up first is being able to set the scale of the scene. So you have your rendered environment, but sometimes you want to zoom in and look in detail at something. So you can take both controllers and you can pinch in to make the scene smaller and expand out to make a scene really big if you want to actually see the details. So moving around, you can, of course, it's VR, so you're in the environment. You can just walk around. But you can also pull yourself through the scene and change your relative position, because you don't have unlimited space. You're still in a VR environment, which is about 6 by 6. There's also D-pad movement. You can aim your controller and use the D-pad, and based on where your thumb position is, it's how fast you can fly through a scene, for example. And that's useful for moving up and down throughout the scene. So one thing that is in VR is just really intuitive is moving objects around. So here's the Vive controller. So this is the D-pad I'm talking about right here. And the trigger for doing most of the animations. These are the infrared sensors that I mentioned before. So basically, right now we only have a few different things that actually allow you to do quite a bit. So you can move objects around and drag objects around by using just simply a trigger based on the position. And then the grip buttons, not commonly used in VR because they're kind of in an odd spot, are right here. And that just resets our scene right now. And I'm going to pass it off to Luciano here.
Before we go into that, because you have probably never seen our software, it looks like this. It has these 4 areas. The bottom area is a gallery, which we can collapse. It's also where we can do some manual measurements and control the editing. Then the main area is actually a viewer, the 3D viewer or 2D viewer if it's a 2D image. And then we have on the top right the display settings, and on the bottom right is the area where we can control what analysis we're actually doing. These 2 big panels plus the one on the bottom can be collapsed, so you get just one big 3D viewer in the middle. To start the VR is very simple. Just there's a button here. It looks like a rainbow. We will hit that button and it will immediately render everything in the VR headset. Here's a couple of movies that we have created with our setup. This is, it shows 2 images because it's rendering for each eye. That's why we have 2 images. So the left eye would be... I'm just going to interrupt really quick. Yeah. The frame rate is atrocious on the other end, at least what I'm seeing, with just by the way. So. Oh, dear. Not very, it's smooth on your computer, not for them. Oh, yes, I know. So I have a very nice, smooth video playing on my screen, but on the screen that we have there in the back, which is basically what you're seeing, because it's another WebEx window open with what you see, it looks a lot more jumpy. So it may or may not look good depending also on your internet speed there. Here's another data set we'll look at today. It's an embryo, and here it's a solid surface. And I'm zooming in. Yeah, I'm just checking there. It's kind of quick, the movie. That's why it looks jumpy. But it's possible to go inside of an embryo like this and look at some of its structure. So right now, I'm going to pause it there so that it catches up. This picture is inside the abdomen of the mouse embryo, and it looks like some of the structure within the lungs that we can see, and I'm exploring that in VR. And I made the embryo so big that it's maybe 2 or 3 times bigger than the room, so I can actually walk inside each of these small spaces, which I've expanded, and explore them. You can see there is one large cell there. This is a 1.4 by 1.4 by 500 pixels, so 1.4K by 1.4K by 500 slices, one channel, and there is essentially one cell in the middle or on the edge of this. To run the recipe is really straightforward. We have to set some parameters, but it's very simple parameters that we have to put in here. Basically the diameter and a little bit about the branch sizes that we expect to have. And then I'm going to hit go. So it says from the beginning, that's applying the recipe. That's what we call the image analysis protocol. And on an image like this, which is 1 gigabyte in size, it takes a few seconds, around 25 seconds. You can see the result will be displayed momentarily on the scene here. It's already loading the result, so the algorithm part has been completed. That's a wheel that shows on the bottom right here. It's gone, and then we will see the result over here, and that's what you already can see in the colored beads. Again, we have a number of ways to represent the results. And right now we're doing it with colored beads, but they could be solid surfaces, or they could be this mesh that you saw on the video a moment ago. We can go into VR by hitting this button here, so switch to VR. And that's what we'll do now on the other computer. So the controls are really very simple, just the two triggers, zoom in and zoom out. One trigger alone is sufficient to grab something, put it back, and like Hoyen mentioned, there is a reset, or Bryce, there is a reset button on the side, which allows us to put the scene back to where it was. So I think we'll move to a different data set.
Yeah.
I think now I'll probably go and show you this data set, and then we'll be just taking questions and discussing things with you guys. Okay? So this is another data set. It is very different from the previous one. It is actually done by done with a technique called optical projection tomography, and it shows an embryo. It was done in Lisbon at the Institute for Science, Gulbenkian Institute for Science. Okay, so here's the data set. It's an embryo. I made it more or less my size right now. So in my view is about 5'10". I can make them first smaller. If I had multiple embryos here, for example, I could have one here, one there, one there, and see all of them and compare them. But in this case, I just have this one single embryo, and I'm interested in looking at what's inside him, looking at some of his structure. So I can make him a little bigger, I can expand him. Now is probably 3 times my size, 4 times now, maybe 5 or 6 times. He certainly doesn't fit in the room anymore. So if you have a bit of vertigo like me, this is...
And you're getting crowded out by the embryo, looks like.
So now he's actually occupying most of the space. That's the hand up there. I'm going to go walk inside. That's kind of the side of his abdomen. And right now, these long structures here, I think they are part of the lung morphology that we can visualize here. So that's this area here. So we are inside the abdomen. That's what we can see. There's some ribs back there that you can also see. And I can walk through and try to find where his head is. Yeah, I'm trying to come back here. Okay. So I think that is backbone. Yeah. So that way, this way is backbone. So I'm going down, and naturally backbone then connects to the tail, and that's where I'm in right now. I'm in his tail. Yeah. And if I zoom this out a bit, here is the tail, and there is the little guy. The eye and the hand, and we can also go inside here to see space. Of course, if we prepare the display differently, we can actually look at different grayscales rendered differently. So we could look at, for example, the bones or some parts of the digestive system appear on their own. Of course, if it's fluorescently labeled with different markers, then we could have that also displayed here. Instead of just one single color, we would have multiple colors displaying different parts. And if there's any segmentation, then you can also display the segmented results. For example, if you have a marker for blood vessels, you can see that separate from other parts of the embryo in this case. So we covered a little bit about the neuron detection that we will launch in April, a little bit about the classification of cells using the machine learning technology that we've developed here, and we did a demo of our VR setup. That's what we managed to cover today. I also mentioned the DRV Beta program, which you can still sign up. Of course, if anybody drops out, you'll be on the waiting list and you can jump in. So you can sign up on our homepage, and it's drvtechnologies.com. And if you have data sets that you would like us to test, we're happy to test them. We can share with you Dropbox or FTP, or if you want to share with us something. We are happy to get that tested with VR or tested on the neurons or the classifier. Anything that you would like us to test, we will at this point. So you can send us data. And I think that's it from me. So thank you so much for joining.
Thank you.
Hope you enjoyed it. And I'll let everybody else say goodbye. Quinn is
Thank you, everybody.
here. And
We're all here somewhere.
Hoyen.
Thank you.
Thank you, and have a good afternoon and a good evening.